Okay, good. Okay, so I'm glad to see that all of you are here. That means you've survived your first week of classes. And more importantly, that means that when you woke up last Monday, you did in fact have an intact immune system, right? Because otherwise things would not be looking so rosy for you right now. Let's start off today by just going over some administrative stuff. So this is the syllabus in La Lima. If you have not had a chance to take a look at it yet, go to the syllabus link and download it, and the schedule, which is what I'm showing here. So here we are finishing week 1. And we're going to get into our discussion of our innate immune system in more depth. And you can see that this takes us through the 5th week of the course, where we have our first exam. All right, and I will talk more about how the exam will work as we get closer to that. On the right side of the screen here, I wanted to show you that this is where you can see the quizzes, which chapters the quiz will be on. So this first week up here, we don't have a quiz, right? But starting the 2nd week, you will have a quiz due. So the quizzes are always due by Thursday night at 11:55 p.m. So essentially Thursday night by midnight. But La Lima is kind of weird because, you know, if you set it to 12 midnight, it actually treats it as Friday. And for some reason, I can't get it to accept 11:59 because I guess it just works in increments of 5 minutes. So remember that. You'll get used to it. So each week, the quiz is due Thursday night by 11:55 p.m. and it's taken on La Lima under tests and quizzes. Right? So it's a timed 20-minute multiple choice quiz. To get you used to that, after lecture today, I will post a practice quiz on La Lima that will be just like the regular quiz, timed and everything, the same type of multiple choice questions, but you won't get graded on it, right? So it's just to make sure that you are comfortable taking the La Lima quiz. And so if there's any technical issues that you have plenty of time to figure it out. Then on Monday's lecture, following Monday's lecture, after we've completed lecture 4 and finished talking about the chapter 2 stuff, that's when I will make the first real quiz available. That will be on chapters 1 and 2, and that will be due then by Thursday night. Okay, so I encourage you between now and then, try the practice quiz first to make sure that your setup works fine with it. And then after you've done the practice quiz, starting on Monday, you will have access to the real quiz due by Thursday. All right. Let's talk more about our innate immune system. Remember, these next few lectures, we're going to focus on the innate side of our immune system and sort of get that out of the way. Because as I mentioned last time, we have a much stronger understanding of how our innate side of our immune system works because it's more or less shared, in conceptually at least, the types of mechanisms with other living things on this planet that are multicellular and have to have some kind of built-in pre-programmed immune system just to be able to keep living. So our innate system is dealing with general everyday responses to the multitude of different pathogens, common everyday pathogens that we're exposed to and have to deal with. The innate system itself has 2 parts to it. One is the immediate response that it has in order to quickly detect that you are experiencing an infection and eliminating that infection very quickly. So our immediate response involves, for example, macrophages that detect bacteria. So here in this cartoon, here's your skin. Maybe you have a cut in your skin. So some bacteria in the shape of these little hot dogs, right, have entered your skin. So your skin, the tissues are being patrolled by macrophages. These are our modestly trained security guards. Right? And so they're on the lookout, and right away, macrophages detect the bacteria, they eat them up through the process of phagocytosis and destroy them. But as they're eating the bacteria, the macrophages are stimulated to start expressing certain genes to make certain proteins that act as small messengers that are released by the macrophage. And these small little messengers are small proteins that we call cytokines. So these cytokines float away from the macrophage and they call for a bit of backup, right? Because the individual macrophage doesn't necessarily know that its job of eating the bacteria, how successful it is in the bigger picture of how much bacteria is actually in there. Where you have a whole bunch of macrophages at work, they're each focused on just eating, eating, eating. So evolutionarily, it's probably a good idea to start calling for backup. It's similar to the idea of the mall cop example that I gave, who is trained to identify a typical pathogen, our rainbow-haired typical clown, right? If, let's say, mall cop on campus, right, one security guard finds a clown, he may arrest the clown, and he may then on his walkie-talkie call back to the dispatch where the other security guards are and say, hey, I just arrested a clown on campus, right? And then the dispatch officer, you know, she might be like, oh, okay, that's good, good job, right? But maybe that's all there is to it. But what if there's a whole bunch of clowns coming onto campus all over? You have different security guards, each sending out these cytokine messages back to dispatch. And so the more messages you get, the more the central dispatch where the security guards are hanging out, you know, eating donuts or whatever, right? They begin to realize, oh, something's happening here. We need to send out some more backup, right? So this is the induced response. As the cytokine messages are being sent out by the macrophages, if those cytokine messages, as they're building up and flowing through your blood, they begin to encourage more of your innate effector cells to come to the area and help out. So this induced response involves the macrophages releasing the cytokines, calling for backup. You start getting inflammation. So this would be, for example, if, let's say, you get a paper cut, right? Tiny little paper cut. You know, we usually don't consider that to be a big deal. A lot of times you go to the end of the day, you don't even realize you got a paper cut, right? And you're like, ow. And then you look and you're like, oh, I got a paper cut at some point, but it's fine. And by tomorrow morning, right, you don't even notice it anymore, right? But, you know, other times, maybe a couple of days later, the little cut is getting kind of reddish and tender and puffy, right? We all know that feeling. And that is the induced response. It still involves your innate side of your immune system because it's pretty quick to react, but this is now the backup coming in the form of other innate effector cells that are flowing through your blood. So this represents a capillary vessel that's really close to where you got the little paper cut, right? So all kinds of stuff is flowing through the blood here, the capillary. And so some of these immune cells will then exit the blood and start helping out. And inflammation is part of this. We will study that in a little bit. So most of the time, this combination then of, even if your paper cut got a little red and tender a couple days later, I mean, you're not that worried about it, right? Because a day after that, it starts subsiding and then you're good, right? Most of the time, this combination of immediate and induced innate responses is enough, right? And you never have to worry about waking up the SWAT team, the adaptive side, because that's the expensive side of your immune system. Let's look at macrophages in a little bit more depth. So this is a wonderful picture on the left over here of a macrophage. This is an electron microscope image of a macrophage, and these little green things are bacteria, right? So you can see the macrophages, it can send out these different pseudopods that can contact and actually bring the bacteria to the macrophage. And here's the cross section of it. So here is some bacteria over here that's been brought to the macrophage, and now it forms a vesicle and it gets internalized into the macrophage, where it then fuses with other vesicles inside the macrophage that contain all kinds of nasty digestive types of enzymes and things that break down and destroy the bacteria. Right? Okay. Consider this though. If the macrophage is just sort of patrolling around in the tissue, then by chance, it will only bump into a limited number of bacteria that are in there. It'd be a lot more efficient if the macrophage knew where to reach out and grab the bacteria. But the macrophage, I mean, it's not smart, right? It's not like it has a macrophage brain or something like that. After all, we're talking about a single cell, right? It's just a bunch of different proteins and other organic, inorganic molecules. So how does it know where to reach? And importantly, how does it actually grab the bacteria? Compounding or making the situation even more difficult is the fact that through eons of evolution, bacteria have developed certain strategies for making it hard for host immune systems to be able to really grab them and eat them. Some of these adaptations have to do with the cell wall of the bacteria. So gram-negative bacteria, such as E. coli, have these very thick polysaccharide coatings on them. So here is a typical E. coli illustration, right? Remember, this bacteria is anywhere from 10 times to like 100 times smaller than the actual host cell in your body. So somewhere around 50 times smaller. Okay, is your typical thing, 10 to 50 times smaller. If we were to zoom in on the coat of this bacteria, we would see that here it has the cell membrane and it has this outer membrane that has these long strands of polysaccharides. So these are chains of simple sugar molecules that are chained together. And it basically creates a situation where you have the whole surface is covered with these lipopolysaccharides, or LPS. Lipo meaning the fact that they are attached to certain lipid molecules that are part of the membrane. And of course, they're long chains of sugars, so they are polysaccharides. So LPS are found on the outside of the bacteria. And what does that do? Well, it's just like a shag carpet, right? A shag carpet, you can really lose things easily, hide things easily in there. And what is it hiding? It's hiding different types of proteins that are part of the membrane to make it harder for the host immune system to detect those proteins. When we first moved into our house in Kailua, the kids' bedroom had this really nice hardwood floor, but I decided one day, I was at Costco and I saw this big like 8x10 shag rug, right? And so it was in one of those big baskets there. So this is when I got to use the big orange carts, you know, at Costco. So anyhow, I bring this thing to the cashier, and she's really funny, right? This lady, she like looks at me as she's ringing it up, and she like scowls at me, like, "Why do you want to buy that?" Right? This is funny, right? Because this is the Costco employee, and she's like, "Why do you want to buy that? How are you ever going to clean that?" Right? And so I was kind of just like joking with her. I said, "Well, it's for my kids' bedroom, and I train my kids really well to clean up after themselves." And she's like, you know, as she rings up, like that I'm giving them $200 for this thing. Anyhow, but it's true, because my kids are always losing bits of Legos and things in the shag carpeting, right? Because the shag carpeting provides a very effective way to hide things. So in the bacteria then, many of the three-dimensional features of proteins and things are hidden from view. So this makes it hard for a macrophage to be able to actually detect and grab onto it. This constant evolution between the bacteria and animals, right, goes all the way back hundreds of millions of years. All right. So how does our immune system overcome this challenge then, right? Let's look at an example and think about this. So here I have, let me stop the sharing for a moment. So here we have, what is this? You would say, oh, it's a beach ball. No, it's not a beach ball. It's a macrophage, right? And so this macrophage is trying to attach to this ping pong ball, all right? And this ping pong ball is a little E. coli, let's say. Now the ping pong ball, it's very smooth, the plastic, so it's really hard for the macrophage to attach to it, right? It just kind of bounces off and there it goes, right? So that ping pong ball is going to make you sick now because it escaped your immune system. Right? So what's one way we could make it easier for this macrophage to hold on to and detect the bacteria? One secret might be you might have certain proteins in your body that you could coat the bacteria with. So in this case, we have a thing here. I can attach it to one of the ping pong balls, put it on there, right? And so now the ping pong ball is coated with this substance. And if the macrophage has certain proteins on its surface that are designed to detect whatever this substance is, right, then the macrophage has an easier time of grabbing onto the bacteria, right? And so what is this substance? It is a very specific protein or a family of proteins in our body that we call Velcro, right? No, we don't call it Velcro. We call them complement proteins, right? I introduced the idea of complement proteins last time. Complement proteins evolved to help our immune system do its job, and that's why they complement. So the complement protein then, the individual Velcro, I think all of you are aware, Velcro is made of two parts. There's the little microscopic hooks, right? And then the fuzzy part that then gets caught in those hooks. So an individual complement protein is just one of those little hooks, right? So just like there are many little hooks and fuzzy things interacting here, right? You would have many of these complement proteins interacting with the bacteria. And so the complement protein, it's a protein, so it's a complex three-dimensional shape. It tends to sort of look like an egg shape. So the textbook just simplifies it as an egg shaped thing. If I hold this close to the camera, I can get it the same size. There we go, right? So this is the complement protein, right? And so in the illustrations in the book, they are portrayed as this kind of blue egg, right? Okay, let's return to our lecture. All right. There we go. So now we're back here. All right, hopefully I didn't screw up our recording here. So complement proteins then are easy to recognize sticky marks that can be put on the bacteria. So complement proteins then are a way to label or tag the pathogen for quick destruction. It's a system of different soluble proteins, soluble meaning these complement proteins flow through your blood in the plasma, and they can coat the bacteria or viruses with a strongly recognized signal that your macrophages have an easier time of detecting. Complement proteins can work in different ways then. They can make the phagocytosis of the macrophage phagocytosing the bacteria much more efficient. They can also serve as a way to send different types of signals that attract additional macrophages to be able to be where the bacteria are. Or complement proteins can act directly to disrupt the bacteria. Because anytime you're talking about little proteins coating something else, if those little proteins can disrupt, for example, the cell membrane of the bacteria, that's another way you can cause the bacteria just to kind of fall apart. So I mentioned that complement is not just one protein, but it's a whole system of proteins. So here I wasn't kidding, right? The book shows them as sort of these blue eggs, but it's important for you to keep in mind, right, that they're not actually blue eggs. They're actually folded up proteins. There are more than 30 different complement proteins involved in our immune system. The most important one is complement C3. In the illustration here, you can see this is a C3 molecule that's floating through your blood. In order for it to work, meaning in order for it to attach to a target, it has to first be activated. Part of that activation involves cleaving it. So you have a pair of scissors here, you cut part of this protein off, and this little part of the protein that floats away, we call C3a, a tiny little piece that floats away and is used as a signal to recruit other macrophages. The part that's left, the main part of the blue egg, we now call C3b. So this is the part that actually attaches and tags the bacteria for destruction. So you can see here it's now bound to the bacteria, right? So this is now putting the sticky Velcro tape on the ping pong ball. This is also a good opportunity for me to warn you that many of the illustrations in the textbook, I mean, they're very cartoony, which is fine because it tries to allow you to see the concept without getting too bogged down with the details, but a lot of times it can be very misleading. For example, in this particular image, it makes it look like the C3b, the individual C3b, is just as big as the bacteria, right? And that's really not true because the C3b is just one tiny little protein attaching to the surface of the bacteria that itself is made up of potentially hundreds of thousands or millions of little proteins, right? So like the little red triangle here on the surface of the bacteria is supposed to represent a different protein of some kind. So in reality, this cartoon blows it out of proportion. Don't be misled by that. The better way to draw it would be the C3b is a really tiny little blue dot, and this whole thing starts getting covered in tiny little blue dots. So how does the C3b attach to the surface? What's activating the complement C3 in the first place, right? And then how does the C3a part that floats away from the C3, how does it actually attract the macrophages? So C3b binds to the pathogen via a thioester bond. Okay, so this is very sort of O-chemy, right? But you don't have to be scared about it because it's not too super in-depth O-chemy. But you would hopefully remember from OChem, okay, that an ester is, you have a group that has a carbon, double bond to oxygen, and then two R groups on it. So if you also have sulfur involved, you would call this not just a plain old ester bond, but a thioester bond. Okay? So in a C3 individual protein molecule, so here again, is it represented more realistically as a folded up polypeptide chain of amino acids. Within here, there's a particular location of a residue. There's a cysteine next to a glutamine, and that creates an exposed thioester bond. Right? Lots of proteins have thioester bonds like this within them, but the important thing to remember is that most proteins don't actively make use of this bond as a way to bind something outside of the protein. So when the C3 is cleaved or activated, it exposes this thioester bond, and this thioester bond then is highly reactive. Okay, because then the shape of the protein is changing, this particular thioester bond is exposed, and it is highly reactive. The part that floats away, the C3a, right, that's this part here from the ribbon diagram of the protein on the left. As a result, that thioester bond can react with things. Now, keep in mind, this whole thing is surrounded by water, right? Because this is all taking place in the aqueous environment of the tissues of your body. So water itself will react with this thioester bond, resulting in this soluble C3b. And if that happens, then this particular molecule of C3b just kind of floats away because it's sort of been like neutralized, right? However, if the activation of that thioester bond happens when the C3 is very close to something else, for example, the surface of another cell, like bacteria, then there is the chance that instead of water getting in there and neutralizing the C3b, the thioester bond will actually attach and react to the surface of the pathogen. And it forms a very strong covalent bond, right? That's practically irreversible. So there are three different ways that the C3 complement protein can be activated by the scissors. We will study all three of these, but we will move through them in different lectures. The three pathways of activating complement are called the alternative pathway, the lectin pathway, and the classical pathway. We will go in more depth to each of these, right? But for now, I want to focus on this alternative pathway. So this is one way where the C3 protein can be cleaved and given a chance to bind to the surface of the bacteria. So the alternative pathway is the preferred first pathway of activating complement proteins that are immediate innate immune system depends on. I will now take you through a series of slides that look really technical and complicated, and there's a lot of molecules interacting with each other. It's kind of unfortunate that this is only like our third lecture and we have to talk about this, but it's important that you understand how complement proteins are activated and how they work as part of our innate system. And so don't let this scare you away. Okay, because as we talk about other things in the semester, there are very few other examples of things being this kind of technical. Right? So step one, let's say in this cartoon here, the yellow part is the surface of a pathogen like a bacteria that is infecting your body. And in your blood, you have a lot of C3 protein, complement 3, floating around. And it's not activated. It's made by the liver and it's floating around in your blood, but you have a lot of it. What happens is that close to the surface of a pathogen, the pH of the area right around the surface of something like a bacteria tends to be lower than the normal pH of your blood. Why? Because, the bacteria is a living thing. It's secreting all kinds of different substances, waste products, and things like that. So the immediate surroundings of the bacteria tend to have a lower pH. And that encourages the C3 molecules that are floating by through there to activate. The first step is water has a hydrolysis reaction that actually starts with the activation. So some of the C3, right, so this is not the cut up C3b yet, will react with water to form what we call IC3. And this means that it has changed its shape a little bit because the water molecule has interacted with it, and that is favored in a low pH environment. So now you have like a hydrogen and a hydroxyl group kind of hanging onto it there to make IC3. Importantly, this reaction, right, to activate the it can happen if the C3 is just floating on your blood somewhere, right, because it's just a water reacting with it, but it preferentially happens a lot more when it's close to the surface of a pathogen because of that environment of lower pH favoring the hydrolysis. So the rate of it happening is much higher next to the pathogen surface than just in the blood somewhere. Right? Step two, here you have the IC3, and because its shape has changed a little bit because of the hydrolysis reaction, it now is recognized by another complement protein called factor B. So this factor B, which is this yellow one here, it now is recruited and binds to the IC3. And when that happens, both proteins change their shape a little bit. Right? So here's the C3 or the IC3, it's a particular shape. Here comes floating by, right, because this is also found in the blood, factor B, and factor B is the right shape to interact with the complement IC3. And when two proteins interact, in order for them to get happy, their shapes change a little bit. In this case, the changing of the shape of factor B, what's the good way to turn this here, right, exposes kind of this cleft in it. And that's what's shown over here, step two. There's a little shape that forms, a little cleft. Right? So we say factor B is a zymogen. A zymogen means it's some sort of enzyme, a little machine that's floating around in an inactive state in your blood until it gets activated somehow. And when it's activated, that means its shape is encouraged to change by binding to something, and now it is in an active shape. This activated factor B, that little cleft in it, recruits this brown complement protein called factor D. And factor D is an enzyme that will then cut the factor B into two little parts. So just with the other complement proteins we were talking about, once it gets cut into two pieces, the little piece that pops off and floats away is now called B little a. The part that sticks around is called BB. This BB part is itself now a very active enzyme. It wants to go ahead and cut other proteins that it encounters. The shape, though, of the BB and the IC3, right, together now we call as one unit. And so we call it IC3BB. And not very original, but it's kind of intuitive. right? That IC3BB shape is the right shape to attract and bind to other complement protein 3s that are floating around, right? And so another molecule of complement 3 comes, binds, and it's positioned in such a way that it becomes what is cut or cleaved by that BB enzyme. And so that BB enzyme cuts that second complement C3, resulting in part of that C3 floating off as C3A, right? And you will have then C3b that's made from it. And that C3b that's made now, once you imagine the dotted line has been cut, the A part floats away, you're left with the C3b. That C3b has now the exposed thioester bond. And if this is all happening really close to the surface of a pathogen, there is a reasonable chance that instead of being neutralized by water and floating away, that thioester bond will now bind to the surface of the pathogen. Okay, so now what happens? So great, you've managed to plop or attach one little protein C3b to the surface, right? And this process is not very efficient, okay? Let me back up, right? Because I got ahead of myself. So step 3 was the factor D, cleaving the B into BA and BB. Right? And now this IC3BB, right, is sort of a enzymatic machine that wants to now work on other proteins and cut them, right? And I said, it turns out it's designed to cut another C3 that floats by. Right? Step 4 then is it will cut that C3, that next C3 that floats along. And as a result, we say that it converts that second C3 molecule into an activated C3b. So therefore, any type of protein complex that can convert the complement protein into an active form, we call it a convertase enzyme complex. So in this particular case, this is a C3 convertase complex, right? Because it's converting a second molecule of C3 into the activated C3b. So what happens now? You've attached one C3b molecule to the surface, right? The next cartoon starts from over here. So right now in this one, the C3b is on the right side of the cartoon. Let's go ahead and put it on the left side as our starting point in the next slide. Okay, so here it is. That's that first C3b that got attached. What happens now is basically a repeat of the previous thing I just told you. It recruits another factor B. The factor B attracts factor D that activates the factor B. It cleaves it into BB and BA. And now this whole C3b-BB molecule that's left, this complex, is now a C3 convertase. And it goes to work just like before. Another C3 floats by, and this convertase cleaves that one and plops it onto the surface, right? And you say, I don't get it. So why is this any different than the previous slide? In many ways, it's not. But the key difference is the previous slide started with the first C3 just floating around somewhere, right? Not necessarily close to the bacteria. So if it starts activating, it's not very efficient necessarily to really be able to increase the amount of C3 coating the bacteria. Right? However, if you start with a C3 that's already attached to the bacteria, and it's doing this process of activating more C3, there's a much higher chance that those activated C3s will just attach to the bacteria rather than floating away. So the location of it now being attached on the surface and going through that same process again really increases the likelihood and the speed of more C3 complements being attached. Because look at over here, this C3b convertase is cutting this next molecule of C3 on here. When that next molecule of C3 is activated and plops down to the surface, what happens to this used convertase complex? Well, it just keeps going, right? So that's why this arrow shows it just is ready to go for another molecule to come. So it's a very rapid feedback loop, because also you have to keep in mind that each one of these C3bs that has been attached now can go through this loop by itself also. So the rate at which more and more complement proteins gets added goes up exponentially, very fast, right? Potentially within a few seconds, the whole surface of the bacteria is covered with complement proteins. Right? We call this process of coating a target, like this bacteria, with some kind of recognizable signal, opsonization. So opsonization means you coat the surface of a target with something. I hope you're thinking now, wow, this rapid chain reaction then, once it covers the whole surface of the bacteria, that's great, but how do you have any control over what's going on here? Because if you think about it, right, the C3b, it's just an activated protein. It doesn't necessarily know what it's doing. Its thioester bond just wants to attach to anything. And if this isn't happening inside of your tissues, then all around this are all kinds of normal cells that you don't want to get opsonized, right? Like your own body cells. So not surprisingly, there are different other proteins that can interact to either increase the rate of this opsonization or inhibit or decrease the rate of this opsonization. One of the examples of a protein that helps to increase opsonization is this properdin, or factor P. The way it works is, you can see in the cartoon, it's this kind of upside down pink L. Properdin recognizes the shape of the C3 convertase complex, and it basically keeps it stable so it doesn't fall apart. And so the more you can keep this convertase complex stable and working, the more successful it is and more efficient it is in converting more and more complement C3. On the other hand, if you have these other factors like factor H might come down and bind to C3b. And when it binds to C3b, it alters its shape in such a way that now C3b is no longer functional and in fact becomes a target of factor I. Right? Factor I, the green one in this picture here, it comes down, binds to the C3b, and it cleaves it, right? It destroys it. So now the C3b has been messed up, and that would stop the process of this C3b contributing to any more C3b being attached to the surface. I mentioned that while this is going on, many of the potential targets of this opsonization are your normal healthy cells that are surrounding the bacteria or just in the same environment. And you want to discourage, right, your body from going around destroying your own healthy cells. So some examples then of factors that are present on the surfaces of our human cells that help discourage opsonization are things like this decay accelerating factor, or DAF. DAF, you can see here you have a human cell surface. So instead of a bacteria, this is just one of your normal cells that was nearby. And unfortunately, right, some of the complement proteins have attached to it. And so there's the threat now that unless you put an end to this, this normal cell in your body might very quickly get coated with complement proteins and get destroyed. So on the surface of your cell, you have this DAF protein that's also kind of bobbing around in the cell membrane. It also recognizes the shape of the convertase, and what it does is it binds to it and it boots off the BB part of it. So it neutralizes the ability of the convertase to work. Another similar factor is this membrane cofactor protein, or MCP. MCP, when it bumps into a convertase, it goes ahead and changes the shape of the C3b to attract that factor I that we talked about in the previous slide. Right? And the more that you can attract the factor I, the more you have factor I neutralizing or destroying the convertase complex as a way to slow down and stop improper opsonization of this healthy cell in your body. So complement, then, to summarize, greatly increases the phagocytic activity and efficiency of the macrophages, right? So by helping out the beach ball being able to attach to the slippery ping pong balls, you really increase the ability of the beach ball to be able to take care of and destroy these little ping pong balls. So the little fuzzy spots then, the little fuzzy spots on the beach ball, what are those? Those are particular proteins that are receptors for the complement protein, right? So the complement proteins have coated the ping pong ball. You need to have something on the surface of the macrophage that is the other part of this, right, to be able to have the Velcro to work. So in our cells, like our macrophages, we have this complement receptor 1 protein. Here in this illustration, here you have a macrophage, and here is the CR1, the blue receptors. So those are the fuzzy spots that are on the beach ball, right? And here you have a bacteria that's been coated by the complement protein C3b. Again, be careful, right? This is not drawn to scale because in reality, if we were to draw this better, the bacteria would just look blue because it's covered with teeny tiny complement C3bs. And so the CR1 receptor, the macrophage, helps this stick together, and it makes it a lot more efficient for the endocytosis process, that is the taking in of something that's stuck to the outside of the macrophage to happen. And because this endocytosis is leading to the ingestion and destruction, the eating of the bacteria by the macrophage, right, we call this phagocytosis. It's brought into the macrophage where it then can fuse with these other smaller vesicles that contain all kinds of enzymes and things that then digest the bacteria. So the effectiveness of this opsonization through complement proteins coating pathogen targets is the reason why most pathogen infections or invasions in our body are eliminated right from the start of infection, right? Remember, I said, even though our sort of intuitive idea of what an infection is has to do with the fact that throughout our lives, we're used to getting little cuts and things that then a few days later start getting kind of red and puffy, and we say, oh, it's getting infected, right? But I told you that in really technical terms, the infection starts the moment the bacteria breaches your skin and gets into your tissues, right? Because technically that's the starting point of when your bacteria is beginning to threaten you and when your immune system is working, right? So it makes sense that that's the moment of infection, right? The inflammation and pain and things like that that we experience later just means that the infection hasn't been taken care of right away. And now it's beginning to present different symptoms that we clinically think of as being, oh, my cut is getting infected, right? Because the majority of the times, those infections are taken care of very quickly, within a couple hours, by complement proteins, helping macrophages identify and destroy the invaders. Okay, so that's the end of the lecture for today. I'll go ahead and stop the recording, and I'm happy to take some questions. Let me stop the recording first.